welcome back. Hi everyone. So today is the season finale. So what do I mean by this? So what I mean is that, as you know, or if you've been following, but if you haven't, so we've been working on this one project. Let me actually just make sure that I'm not streaming. And we've been working on this one project, which was to look at the co-occurrence of antimicrobial resistance genes or antibiotic resistance genes, ARGs, in genomes. And so the goal was to look at that using genomes and metagenomes, and we explored some of the global microbial gene catalog, and we started exploring progenomes. And then in the meanwhile, this other paper came out. Or not a paper, but a preprint, and this was doing pretty much what our intent was here, and so I was rethinking the project anyway. This motivated me to rethink the project. So as I maybe just to be sure, I mean, actually, this extremely open science idea is first and foremost about me trying new things out. So it's mostly and foremost about me keeping learning going. So and that means learning about polars, which we done a tiny bit. I still have not switched for my day-to-day work. I still actually use pandas, but at least 1 hour a week when I'm doing the stream, I'm using polars, and that has actually been pretty helpful. So actually I'm gonna post this on, can I post this on the, I'm gonna post this. Does this work? Or is this now sort of inception? At least 1 hour a week. Okay, let's avoid that. So I'm gonna post here. So this is this paper. So when we went through some of the figures before and showing even how some of the figures are pretty similar to the figures we have been generating. I mean, obviously more advanced, obviously more polished, but these were the figures that we were looking at, exactly this type of heat map, these type of heat maps, and so I've learned polars. I've also and this is another of these ideas of this project, was that I would be using some of our own tools, and because that's one of our principles. And again, I'm going to be posting these in the chat here. So we have this manuscript. Manuscript that I wrote. It's a slightly different type of manuscript. It's an opinion piece about the principles we use for how we develop software, and one of them is this practice of so-called dog fooding, which comes from this expression, we eat our own dog food. So we develop tools for others to use. For example, in this case, we develop the ArgNorm tool, and I developed Jug, and so I use them myself. So actually, in this case, Jug was a tool that started as people call a scratch your itch tool. So it's something I really needed on a day-to-day basis, and so I developed it, and I actually developed it for a very, very long time, when I think I was literally the only person using it, and I wasn't even thinking about whether others would be interested, because actually it started fairly ad hoc. It was not, and with time it became more structured. It became better code, and at some point I thought, I'll do a bit of advertising about this. I think I've mentioned it before. I think the tool has not really received the amount of interest I had hoped, and it's always a little bit of disappointment for me, because I do think that was one of, in the end, not so much through very clever design, but through iteration, it did become something that's very, very useful. So but that's all, eating our own dog food. So I use Jug, I developed Jug, and I was the first user. In the case of ArgNorm, actually a little bit, there's a similar story in a very different context, but we actually did start out using it only internally. It was a side project with the goal of eventually, using it on a comparison of methods that we're still working on, actually. So that's actually still also a live project, but we realized that the tool itself could be useful, and we realized it because people were using it, and so somehow that was maybe not 100% surprising, but it was at least a little bit surprising. And so we decided to work on the tool, tool, and polish it, and so this has been very helpful. This idea of we eat our own dog food for me as well. I think we found some of the ideas of the improvements from ArgNorm have actually come from me here on the stream, looking at the issues. Right. So what did we actually do? So this is this is now a little bit. So we developed this co-args project, and in the co-args project, so now this is a little bit. There's a little bit of some dirty files here. But let's see. Right. So I'm showing what we did at the very, very beginning. Okay, so let's just pick. Just see all tools, but let's use deep arg. So we built these functions to load the data. Then I'm trying. Interesting. Now remembering everything that we did here. So okay, so these were these abundance abundance tables where for each sample, so in sample being a metagenome, and each potential gene in a human gut sample, we had this count. So we had this number that was normalized, and then most genes are very rare. Most genes are rare, in fact, and actually antibiotic resistant genes are less rare than others. But we're not interested in the genes that are very rare. They just don't have a lot of information with respect to co-occurrence. They just, that's just impossible, right? If a gene shows up in 5 samples, it really cannot. There's not a lot of information there. At best, if it co-occurs in 3 samples with something else, is that informative? It's very hard to say. On the other hand, genes that appear in every sample, so genes that would be constitutive genes, constitutive not for each individual genome, but constitutive for a community, because we actually know that there are these core genes that are core in the microbiome, but they are not core in genomes. So that means that there are genes that are rare in genomes and frequent in metagenomes. So this means that there's some niche there. This is one possible interpretation, is that there's some niche there whereby there will always be some microbe performing that function, but it's always a single. It's not a but probably it's not a very widespread function. So and this is by contrast with, say, with say, basic machinery of life functionality, where it's common in every genome. So anything to do with DNA replication of it's going to be present in every genome. And then since it's very common in genomes, it's very common in metagenomes. Whereas if there are other functions where they're uncommon in individual genomes, but then in a community there will always be at least one microbe there that's going to perform that function, right? And that's also perhaps not the most interesting, because again, if it's a bit the mirror image. If something is present everywhere, at the extreme, so let's say something is present everywhere or almost everywhere, we also don't get that much information about it, because it's going to co-occur with everything else, right? If something's present. So what we wanted was something that was present somewhere, frequently enough, but not too much. And so we decided here that we were going to say the minimum is 20% and the maximum is 80%. So something between 20 and 80%. And we estimated that or computed that by taking the number of samples, dividing by 5, and then and then that's the minimum size, and then the number of samples minus that would be the maximum size. So we filter the columns, and let's see, yes, so about 7,000 samples. So this is the number of human gut samples in the GMGC. We started with 600 genes, and out of that 162 were in this 20 to 80, and now let's just count all possible co-occurrences. So this is probably not the cleanest code, but it works, and it works fast enough. Oops, sorry. So we had run this. Oh, I skipped them. Okay, let me do this again. Okay. Now, I remember this. So we need this out. We need this clear, because first we compute this. All right, there it is. Right, and then we had seen that we have these groupings. So here we've used clustering to reorder the matrix so that the groupings become apparent, and we have these groupings, and then we saw that this was pretty common, and actually if we go to the right, they use a slightly different layout, and I guess they use a different diagonal, or but again you see these groupings, these squares in the matrix. Right. Okay, I guess they didn't order these in any particular way. But you can see, so here, this is a little bit more advanced. They have, because they have some modeling beneath this, which we don't. Right, so that's what we did. And then we sort of switched stacks very drastically. So from the beginning, this idea that we wanted to have these two views of the problem. So the metagenomic view, where we look at the community as a whole, and the genomic view, where we look at individual genomes. And these are different questions, and so to look at the metagenomics view, we started looking into progenomes. So we had progenomes, and so we discussed a little bit how progenomes was this, or is actually this database of genomes. They've been clustered using SpecI clusters, which is a genomic definition of species. So this is our species defined by the by clusters of specific genes, specific marker genes, single copy marker genes, and we could download this Beckeye clustering table and then download genomes and then run RGI on this. So an RGI is the tool developed by the CARD database, or not developed by the CARD database, developed by the same group that develops the CARD database, and that maps genes to CARD database and to the arrow, which is the antibiotic resistance ontology. So here, we saw this, we solved a lot of little problems with file formats and with loading the TSV files with polars. And I mean, this was also perhaps one of the goals. I mean, maybe a slightly less selfish goals than the previous ones I discussed, where I wanted to show this side of things, especially if someone's watching who doesn't have as much experience, that you see that I have a lot of experience doing this type of stuff. However, even with this much experience, it's still the case that we spend a lot of time really just solving technical things that have nothing to do with the biology, have nothing to do with the scientific question at hand. They have everything to do with why this in this case, so there's this column called nudged, and nudged is a Boolean column, except that for the most part it's empty, and so it's an NA or NaN or none column, except that it's in some cases it's completely empty, right? So in some genomes all the outputs have an empty nudged column, and so when Polars tries to read that, it cannot guess because how could it frankly guess that nudged should be Boolean, and so in some cases it would load the table as Boolean correctly because there will be some true or false values in that column. In other cases it would just guess that it was potentially a string column because that's the most general guess, and then later this led to issues when we tried to concatenate them. Right, but we did. So here we had to solve this, and actually we spent quite a lot of time solving this. And again, or not again, but another very common feature of these things is that this cost us a lot of time, and then in the end, the fix was this little thing. So this is so this is about 22 characters, and so this 22 character fix can easily cost you a lot of time. So this is very typical, and I wanted a little bit to demonstrate to especially people who are starting out computing more. I think if you're coming from a different background, sometimes you think that the hard part is, or you underestimate, to borrow a word, how much time you end up spending on these very tiny technical issues that are conceptually very trivial, and in fact once you figure them out, they're also very trivial to fix, but figuring them out is where you spend your time. I mean, this is at the end of the day, this is the job, is to figure out these million of these little tiny things, right? And so we ran this. We chug, and so I showed how we've actually only done a tiny part of this. yeah, so we've run less than 1% of the whole thing, but we wrote the code, and we could have, if we wanted to continue with the project, we could have now continued. So everything. And then the goal. So minus capital S minus capital R is going to not just show us the files without wrapping, so that we have to scroll right to see the outputs. Right, so here's the output file. So here we have some. So this would be all of the outputs for, what was this? SpecI 23, which I don't know what it is. Out of curiosity. Okay, it's mostly a Streptococcus, some Streptococcus suis. Interesting. All right, so it says 137 genomes, 2,338 biosamples. Maybe. So here, actually, now that I was okay. Okay, interesting. 1307. Okay, I'm rethinking a little bit what we did before. Because before we were treating, or the code was treating, so let's back up a bit. Right. So this is the list of all of the genomes. But you can see that it's something dot something. and I think that this first half is a tax ID, and the second half, this is a biosample ID. And so now I'm thinking, so before we were just treating these as, oh, look, there's a, this is a genome. But now I am thinking that, in fact, we should have maybe only gotten one single genome from each. So this, these two we should not have treated. So we should have treated this more carefully. So probably consider that these two are sequencing more or less the same thing. Not sure. Okay. I can actually check. Okay, this is interesting. Okay. All right, I'm going a little bit on a sidetrack here, but I wanted to check this. Okay, so all right, so a little bit here, backing up. So I was noticed this that on the website, so Progenomes was making this distinction between genomes and samples, and so 137 genomes and 2,000 samples, which we hadn't done before. So my interpretation is that this means that this was basically the same genome, so probably this is actually a reference strain, and it just got sequenced multiple times, and probably each one of these is going to have its own little variations, but it's going to be sort of SNP level variations, very small changes, which are incredibly important for certain studies, but maybe not so much for what we're doing here, where we're looking for sort of larger scale structure variation, so presence of new gene cassettes, absence, that kind of thing. And so just as a sanity check, first I did this. So just looking at how much is reported by RGI, and you can see that, so this 1307, 184, 185, 185, 185, let's ignore those, 186, 183. So it seems to have 185 genes, except that maybe some assemblies are a little bit incomplete. Maybe this one is a little bit more incomplete. And then this one has several more, which is maybe a little bit surprising. But then this one is very different. So this one has, a good, this one has a good 30 genes more. So this one is, so looking a little bit at this. So let's just, okay. No, wait. Okay. Okay. I'm sort of slowly getting the hang of this polars. So what would I want to do now? I would want to... What would I want to do? I would want to check whether... this is actually where a little bit of scribbling on a piece of paper helps. So I'm going to take out a piece of paper. Draw something. So what I want to do is I want to take all of the arcs Alright, so actually, okay, let's see. Does this work? Okay, this works. All right, so here's one thing I could do. I'm gonna build a 10 by 10 array. Okay, I'm switching to writing this not on the command line, but on an actual file. And now let's see, okay. So we have G and G2. Does this work? Yeah. Does this work? Oh. Okay, this is okay, this is actually a little bit okay. All right, so the first thing, which is a little bit surprising, but I think it makes sense after I saw it, is that right, so for example here, let's say for this G, which is G is whatever this is. So this is 182, which is the size I expected, but apparently, so the size of the set is smaller, which is not unexpected. Not unexpected once you think about it, but, well, I guess it's literally unexpected, as I say. So once you think about it, it's easily explained, but it was a little bit unexpected for me. But then I wanted to see what was, so 111 is sort of, is how many genes this has, this last, and then, so the diagonal is how many genes that particular genome has, and then how many it has in common. As you can see, in some cases, it's basically almost everything. So for example, this and this. So you can see this. So this has 111 genes, 109 in common with this one, 109 in common with this one, all in common with this one. And then, but then there's slightly lower numbers. But the top one that has 120 genes has relatively less of them are shared. Okay, I'm not sure. And the top one is also the one that is a different genome. I'm not sure that there's much to be learned from this. Okay, yeah. All right. In any case, well, what's the terminology? Biosample? Okay. Just so yeah, so not sure exactly what this means, but oh, I'll do that later off stream. Okay, so yeah, so we got a little bit nerd sniped here with a slightly different issue, but as we keep it's mostly the day-to-day is mostly a collection of these things, sort of running into random issues like this. Yeah, let me just running into random issues like this, and so I'm actually maybe just cleaning up. Let's just clean up a little bit here. That's something completely different. That has to do with probably something for my kids at school. I think that was math class registration, sort of. Not sure why I had the, not sure why I put it there. All right. So yeah, I'm not. Right. So all right. So as I said, I think now probably next week we're taking a little break. There's actually a conflict at this exact time, so it actually matches correctly. I will do some updates on the mailing list as well. I have an idea for a slightly different project that I still want to be working on. I I still think this is a pretty decent setup. I'm not sure how it's not exactly it's not exactly thousands of people. I'm not quitting my day job, but I'm mostly doing this as a way to learn new things, as a way to keep myself a little bit apprised of things like Polar seems to be the new kid on the block in that space, and trying to keep up with that. Keep using our own tools, keep eating our own dog food. And so from that point of view, it was a big success, this project so far. And so with that, I am going to, so I should figure out how to do this a little bit better in terms of streaming when I'm at home and I don't have multiple monitors. But actually maybe next week I'll be back, so next week we skip, but then in the following week we'll be back in the office with a whole new series, a whole new project, and extremely open science season 2. So with that,